Welcome everyone to this week's edition of the Extracellular Vesicle Club. Thank you all for joining. So our guest moderator today is Olesia Gololobova. Olesia is now a postdoc in my lab and she's arranged this event today. So Olesia, thanks so much for co-moderating, and I'm going to hand it over to you.
Thank you, Ken, and it's my pleasure today to introduce Dr. Sara Busatto. She will present an amazing paper with very interesting animal model about extracellular vesicles derived from brain metastasis. Welcome, Sara, and please go ahead.
Welcome, everyone, and thank you very much to Drs. Whitwer and Gololobova for the invitation. It's a pleasure being here today. And thank you for attending this EV Journal Club. My name is Sara Busatto, and to date I'm a postdoc at Boston Children's Hospital and Harvard Medical School. But today I will discuss the results from this proof of concept study that I did as a postdoc in Dr. Wolfram lab at Mayo Clinic in Florida. This work was published in 2020 in the Journal of Nanobiotechnology. The work is titled Brain Metastasis-Derived Extracellular Vesicles Induce Binding and Aggregation of Low-Density Lipoproteins. So the main topic that we will discuss today is cancer metastasis. Unfortunately, cancer metastasis cause up to 90% of cancer-related death. This is because many aspects of the metastatic process are still poorly understood. In fact, we have limited early diagnostic options and also limited available therapeutic strategies. In particular, today we are going to talk about brain metastasis. There are three common types of cancer that often result in brain metastasis, and I'm talking about lung cancer, breast cancer, and skin melanoma. The diagnosis of brain metastasis determine a poor prognosis for the patient. If we take into consideration breast cancer patients that are diagnosed with brain metastasis, their median survival time after the diagnosis is 10 months. And if we talk about melanoma patient instead, the patient diagnosed with brain mets have shown a response rate to available therapy, which is just of 5%. Also, in the talking about brain metastasis, we don't have any early diagnostic option. Usually they are diagnosed with imaging techniques, meaning that the secondary tumor is already formed and active. And about the therapeutic option, there are few therapeutic options available, but overall, we have very limited therapeutic strategies. Interestingly, in the past years, it has been shown that extracellular vesicles play a key role in the metastatic process, and in particular in brain metastasis preparation. Extracellular vesicles support the metastatic process in the early and also late steps. Extracellular vesicles are secreted by primary tumors, are then emitted into blood circulation. They travel both short and long distances. They target new healthy organ where they modulate the existing microenvironment, creating a pre-metastatic niche, which is a favorable microenvironment that will then support the onset, the survival of cancer cells and the onset of metastatic lesions. Then also the micrometastatic lesion will secrete their own EVs that then will support a full functioning metastatic colonization. In the literature, we can find numerous informative papers that studies the interaction between cancer-derived extracellular vesicles and different type of target cells that we can find both at the primary site and at the target site. And I'm talking about endothelial cells, epithelial cells, and resident immune cells. Speaking of the interaction between cancer-derived extracellular vesicles and endothelial cells, which are the main components of the blood-brain barrier. In 2018, Dr. Morad, mentored by Dr. Marsha Moses, they published a fundamental paper in ACS Nano showing how brain-seeking EVs derived from brain-tropic breast cancer cells are able to promote an increased incidence of brain metastasis. In particular, a murine model preconditioned with repeated intravenous injection of brain-seeking EVs developed more brain metastasis compared to murine model preconditioned with extracellular vesicles derived from parental breast cancer cells, MDA-MB231. This is because the brain-seeking EVs are able to interact with the blood-brain barrier. Specifically, after a few minutes after their injection, systemic administration, they breach the blood-brain barrier and they are uptaken by the astrocytes. With their passage through the blood-brain barrier, they cause a decreased protein level in endothelial cells of RAB7A. RAB7A is an endolysosomal protein, which is linked with cell degradation pathways. They leave instead unchanged the level of RAB11 protein, which is instead an endolysosomal protein, which is linked to recycling pathways. Overall, these results may lead us to the conclusion that brain-seeking EVs are able to modulate the metabolism of brain microvascular endothelial cells, switching their metabolism from degradation pathways to recycling pathways and promoting their increased transcytosis through the blood-brain barrier. So they are able to breach the intact blood-brain barrier. As I said, we can find this and many others informative paper about the interaction between extracellular vesicles and target cells, but we tested another complementary hypothesis as we realized that there is not a lot of focus on the interaction between cancer-derived EVs and circulating components. In fact, in the majority of the studies out there that analyzed extracellular vesicles, but biological nanoparticles in general, they are all taken as a single subclass of nanoparticles and studied alone. Although this is unlikely because we know that both eukaryotic and prokaryotic cells produce various type of biological nanoparticles that overpopulate the biological fluid. These nanoparticles also are known to overlap in physical properties such as size and density, and they are also involved in transport and communication process. If we take as an example the blood, in blood we find mainly extracellular vesicles, but also all the subclasses of lipoproteins and other globular protein carriers such as albumin and transferrin. In particular, extracellular vesicles and lipoproteins share similar size, density, structures, and sometimes they overlap also in function. Interestingly, lipoproteins are another subclass of biological nanoparticles that play their own role in cancer spread and metastasis. In particular, low-density lipoproteins. But let's see the general topic as how lipoprotein play a role in cancer. So we know that cancer patients often have also lipid and lipoprotein disorders. We also know that cancer tissues, in particular lung cancer, breast cancer, and melanoma, overexpress LDL receptor, meaning that they gain an advantage by the uptake of circulating low-density lipoproteins. Talking about breast cancer patients, we know that obesity is a risk factor for breast cancer patients, in particular if we talk about postmenopausal patients. And obesity is again often characterized by abnormal lipidic and lipoprotein profiles. In a study published in 2016, it has been shown that triple negative breast cancer cells, MDA and B231, if incubated with specifically low density lipoprotein, they exhibited increased migration, invasion, and proliferation abilities. Contrarily, if stimulated with high density lipoproteins, they didn't show any specific variation. In the same year, another paper published the first evidence that vesicles and LDL may interact. After that point, other reviews and papers discussed how LDL act as a fuel for cancer spread and metastasis, and how both EVs and LDL are involved in the tumor microenvironment, and specifically in the adaptation of cancer cells in the tumor microenvironment that usually is hypoxic and acidic. Last but not least, similarly to extracellular vesicles, also low density lipoproteins can be transcytosed through endothelial barrier. So another very interesting circulating components that we can find in blood, and that is deeply involved in the brain metastasis, in particular in the formation of the pre-metastatic niche, are bone marrow derived monocytes. In physiological condition, bone marrow derived monocytes are exchanged at low rates and mainly at the blood cerebrospinal fluid barrier in the choroid plexus. But the situation changes during pathological processes. In pathological processes, bone marrow derived monocytes enter the central nervous system. And in particular, we know that other than brain resident microglia, the bone marrow monocyte derived macrophages represent the most abundant non-cancerous cells that we can find in the brain tumor microenvironment, both of primary and secondary brain tumors. The presence of brain tumors, we know that induces significant recruitment of myeloid cells, macrophages, monocytes, and granulocytes. Unfortunately, so far, little is known about the mechanism that drive and allow the passage of blood cells, leukocytes, through the blood-brain barrier. The literature hypothesizes that it could be by paracellular transport and transcellular transport. So far, a few factors like chemoattractant protein 1 and granulocyte macrophages colony stimulated factors may play a role in the migratory abilities of myeloid cells across the blood-brain barrier. In order to study our hypothesis, namely that cancer-derived EVs interact with circulating components, we chose two models, two brain metastasis model. One was from breast cancer, and we isolated the EVs both from regular cancer cells, MDA and B231, and from the brain-seeking counterpart, isolated in vivo by the Massaguet lab after multiple cycles. And the Massaguet lab was also the lab where we purchased the cell line. The other model that we chose was melanoma. As a regular cell line, we used the 8375 cell line, and as brain metastasis cells, we used a cell line established from the Sarkaria lab in Mayo Clinic, Minnesota. The cells were taken directly from brain metastatic tissue of a melanoma patient that underwent surgery. All the EV samples from the 4 respective cell lines were isolated using the tangential flow filtration technology. It's a technology that couples a tangential flow with filters with the design with pores of different sizes. So you can customize and decide a different size cutoff. Compared to the other classic used EV separation methods, TFF has an increased yield and also purity, at least towards the protein that do not overlap in size with the size cutoff that you decide for the EV separation. After the separation and the concentration of our EV sample, we also performed a thorough and necessary characterization of our samples, comparing side by side the regular EVs and the EVs released by the brain-seeking cells. For the breast cancer cell lines, both the regular and brain-seeking samples showed that a nano-sized population of extracellular vesicles enriched in characteristic EV biomarker and displaying a negatively charged surface. We checked and analyzed the morphology of those samples using two techniques, namely the cryogenic transmission electron microscopy and the atomic force microscopy. In this case, we decided to show the cryo-TEM images, where we can appreciate both unilamellar and multilamellar vesicles, consistent to what has been showed also by other papers in the literature that analyzed extracellular vesicle samples with this microscopy technique. Talking about the melanoma extracellular vesicles, again, also here, both the regular and brain-seeking EVs were nano-sized and rich in characteristic biomarkers. And this time we can appreciate the morphology analyzed by atomic force microscopy. The EVs have a round-shaped morphology and are deposited on a clean background. As a first step, we wanted to corroborate the interaction between cancer-derived extracellular vesicles, regular and brain-seeking, and an in vitro model monolayer of brain microvascular endothelial cells, as they are the major components of the blood-brain barrier, at least on the blood side. So we established in our lab this in vitro model of brain endothelial cell monolayer, culturing primary human brain microvascular endothelial cells for 10 days in order for them to form junction, both adherent and tight junctions, characteristic of the BBB structure. We checked the biomarker expression, both by confocal, here you can see the expression of zonal occludence 1, and by Western blot analysis, here you can see the expression of N-cadherin, BE-cadherin, Claudin 5, and occludin. So we consider this as a simple yet reliable model of brain endothelial cell monolayer. We tested our monolayer with our two different samples of EVs. First, we performed a time-dependent uptake experiment, but we couldn't detect any difference between the two EV formulation. The two EVs didn't show any specific cytotoxic activity towards the cultured endothelial cells. Then we also seeded the human brain endothelial cells on a transwell assay, and we treated the cells with the two EVs separately. And then we tested whether the EVs increased the leakage of the endothelial cell monolayer by incubating the monolayer with different protein and dextrans covering a broad range of molecular sizes. We can conclude that both EV samples display a moderate effect towards the endothelial cell monolayer permeability, although there was no difference between the two groups. So the moderate effect was displayed equally by regular EVs and brain-seeking EVs. So we decided to test our hypothesis that extracellular vesicles may also interact differently before reaching the target sites with other circulating components For example, lipoproteins, and we took into consideration the two main subcategories of lipoproteins, low density lipoproteins and high density lipoproteins, and the already discussed monocytes. We started with the low density lipoproteins. We wanted to test whether EVs and LDL first interact. So we mixed a fixed quantity of LDL with a fixed quantity of EVs. After a passive mixing, we wanted to to determine the presence of CD63 in the native and mixed sample. CD63 is a transmembrane protein characteristic of the extracellular vesicle population. And as you can see here, the sample of EVs mixed with LDL didn't show any signal for the cluster of differentiation 63 protein, meaning that suggesting that the LDL may coat the extracellular vesicle structure. Analyzed by nanoparticle tracking analysis, the brain-seeking EVs mixed with LDL was the only sample showing a significant decrease in particle count number compared to the other samples. So we wanted to further test this possible interaction by running our samples through size exclusion chromatography column. We chose a commercially available SEC column that declares the ability to separate and elute at a certain fraction EVs, which are separated from other possible biological nanoparticles, among which we can find also lipoproteins. The company declares also that lipoproteins are retained into the size exclusion chromatography column and eluted at different fraction compared to EVs. After processing all our sample through size exclusion chromatography, we wanted to detect by an ELISA assay the presence and the levels of apolipoprotein B, which is the main protein component of the low density lipoprotein. As you can see, the native EVs, both regular and brain-seeking, showed a negligible amount of ApoB in the sample, whereas the sample of EVs mixed with LDL showed, of course, a considerable amount of ApoB in the sample. What happens if we run our sample of EVs mixed with LDL through size exclusion chromatography? That the ApoB level goes back to negligible. So effectively, the lipoproteins are retained, are not eluted in the fraction where we expect to have the EVs. Analyzed by nanoparticle tracking analysis, the EVs eluted through SEC showed a considerable amount of particle per ml, whereas the EVs mixed with LDL and run through SEC, they showed a significantly decreased number of particles. If we want to dose the presence of CD63 by an ELISA assay, again, the sample of EVs mixed with LDL and run through size exclusion chromatography column, they showed a non-detectable level of CD63 protein. Altogether, these results suggested to us that EVs bind to LDL and form a stable interaction. We also analyzed the interaction between extracellular cancer-derived extracellular vesicles and high-density lipoproteins. Again, as before, we mixed fixed quantity of high-density lipoproteins and EVs. And after a simple passive mixing and incubation, we perform an ELISA assay for CD63. As shown before, the mixed sample of EVs plus LDL didn't show any signal for CD63, suggesting again a possible coating of HDL on the EV surface. However, once we ran our HDL EV samples through the size exclusion chromatography, we saw a decrease in particle count, although not as significant as with the LDL samples. And if by ELISA, when by ELISA we analyze the presence of apolipoprotein A1, which is one of the main protein components of the high-density lipoproteins, we saw that the HDL marker was just detectable in the EV and HDL sample pre-size exclusion chromatography, but not in the one that were processed through size exclusion chromatography, corroborating that HDL are not eluted at the same fraction as EVs. Contrarily to what has been shown with the low-density lipoprotein, this time, if we analyze the fraction of EVs mixed with HDL and run through SEC column, we could detect this time a considerable amount of CD63 signal corresponding to EVs, meaning that although the EVs may interact with HDL at the beginning, similarly to what they do with LDL, their binding is less stable and is compromised by the SEC, which is run by a simple gravitational flow, because after the SEC, we have back our single and detectable extracellular vesicles. We wanted also to check the morphology of our sample pre and post incubation with low density lipoproteins. In the left panel here, you can see the morphology of the samples alone, namely regular extracellular vesicles, brain-seeking extracellular vesicles, and low density lipoproteins. Once mixed together and imaged again, we see two different type of morphologies. In the sample of regular extracellular vesicle mixed with LDL, we see random association between the two particles. But if we look at the cross-section graph here, the blue curve represents the regular EVs plus LDL sample. We have a flat background and the single particle profiles. If we look at the brain-seeking EVs mixed with LDL instead, we saw bigger aggregates that measure few hundreds of nanometer. And if we look at the cross-section graphs, the orange line here, it represents the aggregate at the end that has different heights. The lowest one is about 10 nanometers, and it goes up to 25 nanometers thickness. So these results suggested to us that although both cancer-derived EVs interact with LDL, the brain-seeking EVs, the brain-tropic EVs, are able to cause pronounced LDL aggregation. Looking for some physical structural difference between the two EV samples that may somehow explain this different interaction between brain-seeking EVs and circulating low-density lipoproteins, we tested their composition in lipid phases by using lipophilic probes that are known to be to display different affinity based on their structure towards different type of lipid phases. Once we tested the intercalation of our EVs with a long chain octadecyl lipophilic probe, which is known to display high affinity towards lipid gel phases, the brain-tropic EVs, both from breast cancer and from melanoma, display a significantly higher intercalation with the probe, meaning that the brain-seeking EVs are maybe enriched in lipid gel phases, which are known to be ordered and formed by tight lipids packed together along on the length scale. Contrarily, if the two EV population formulation were compared for the labeling with other probes, such as the cholesterol body pie, which is more fine towards lipid rafts, the two EV formulation didn't show any specific difference. Of course, this is just a very preliminary experiment and is a proof of concept study, but this may explain the different interaction with the lipoproteins that perform mainly lipid transport. So after testing the interaction between EVs and circulating components such as LDL, we found very informative paper in the literature saying that LDL, specifically the aggregated LDL, are more uptaken by circulating monocytes, and that the monocytes are involved in the preparation of the pre-metastatic niche in the brain. So as a last experiment, we wanted to study the uptake of native EVs and EVs mixed with LDL, and their uptake from the circulating monocytes in culture. Both breast cancer and melanoma brain tropic EVs, when mixed with LDL, showed a significantly increased uptake from circulating monocytes compared to the native brain tropic EV samples, but also to the regular EVs that didn't show the same pattern. As a control, we look at endothelial cells, and there we find out the totally opposite pattern. So if the EVs are mixed with low density lipoproteins, their uptake is decreased, and is decreased equally between regular EVs and brain-seeking EVs. So taking all together, all the results of this proof of principle study may lead us to the conclusion that brain-seeking EVs, low-density lipoproteins, and monocytes may interact between each other and play together a role in the preparation of brain metastasis, in particular in the preparation of the pre-metastatic niche. In conclusion, just a few points to summarize all what I said so far. From this work, we can say that other than EVs, also other biological nanoparticles that we find in blood, can find in blood circulation, such as low-density lipoproteins, may play a role in metastasis. We can also say that cancer cell-derived EVs associate with the LDL structure, and their interaction are not equal among all the cancer-derived EVs, but they have different interaction, which are dependent on their pro-metastatic potential. Brain-seeking EVs are able to cause LDL aggregation and also an increased uptake from the monocytes, which are components of the brain pre-metastatic niche. Brain-seeking EVs are also showed to display higher affinity towards long-chain octadecyl probes, lipophilic probes, which are known to integrate more in gel phases of lipid bilayer, indicating that these or other may be differences that play a role in the EV membrane structure and structure properties, and these characteristics may play a role also on their pro-metastatic potential of the EV themselves. And these studies provide also the first indication that in a cancer metastatic context, EVs interact with LDL, and they may play a role potentially modulating the monocytes and play also a role in the overall metastatic cascade. Before the Q&A session, I would like to thank very much my former lab and my former mentor, Dr. Joy Wolfram, and all the colleagues.
Thank you, Sara. It's a great talk. We have a few questions right now. Let's start from question from Lucia. Do monocytes show different phenotypes, M1 or M2?
We didn't test So the mechanistic study and the biological effects that EVs may have on monocytes other than uptake, they are ongoing. We didn't test the change in phenotype, but we are testing right today. What they are testing in the Wolfram lab, the changes in the secretome after the uptake of different type of metastatic EVs.
Yeah, because we did publish that cancer-derived EV are taken up by monocytes, and there are changes in the M1, M2 phenotype, but we didn't look for LDL, for the presence of LDL in that case. The other question I had was about the monolayer assay, where you did analyze dextran or other, different sized particles. So how did the monolayer behave? Because we have seen different things depending on what cancer cell EV we add on the monolayer.
So again, we added our EVs just for 24 hours on the form monolayer. And after 24 hours, we could appreciate from caused by both the metastatic EVs, a moderate increased leakage. But again, we didn't see any difference that could explain the in vivo results published by my group now that showed how the once injected in vivo, this EV effectively promoted the formation of brain metastatic lesions. So since we didn't see a difference there, we looked on for something else.
Yeah. Okay. Thank you, sir. Okay. Nice talk.
Thank you.
We have another question about separation. Jan, would you like to unmute yourself and ask this question?
Hi, yes. Hi, Sarah. Thank you for the nice
Hi.
talk. Yeah, I just had a question about the observation you have that in your case, SEC removes these complexes. So if you incubate your EVs with these lipoproteins, you see that SEC removes these complexes, but normally SEC only removes smaller particles, right? Because they can enter the sepharose, the 75 nanometer pores, and all the bigger stuff is expected to elute out.
Mm-hmm.
But in your case, you don't see this. So I'm just wondering, how do you explain?
Well, I didn't test the previous or later fractions. I just tested the fraction where I'm supposed to find the EVs as declared by the company. So I cannot say that my complexes are retained by the column, or I cannot say in which fraction they are eluted. I can say that they are not eluted together with EVs, or if they were EVs. Mm-hmm. So probably the fact that there is let's say, if you want to call a protein corona formed by LDL on the top of the EV structure, these play a role in their processing through the SEC column. But without analyzing the other fraction for ApoB or other biomarkers, I cannot.
Yeah, but I mean, with this type of small columns, normally, the EVs are the first thing that elutes because they are bigger than the 75 nanometer pores of the sepharose. So these are in the first fractions where you find something are the EVs, and everything that is bigger than 75 nanometers. So
I also read in the company website that on the top of the column, there is the filter that is supposed to stop all the lipophilic and lipid-based structures. So I don't exclude that maybe they don't even run in the column and that they get stuck in the filter. Yeah,
okay, that would maybe make sense indeed. But then, just a follow-up question. Many publications, I think, have shown that SEC is actually quite efficient to isolate EVs from blood plasma. So if these complexes would be present in blood plasma, which you would expect, right? Because there's a lot of VLDL there.
Well, so far we just showed that the complex happens with a certain type of metastatic EVs, so it depends.
But also with the regular EVs, right? So with the regular EVs, it was the same thing.
There are association, but not aggregation, yes.
But after SEC, you also saw that, there was a lot less concentration of the regular EVs when they were.
But the regular EVs are still cancer. They are not just brain targeted.
Yeah, okay. But in any case, like when you isolate, from cancer patient plasma, usually SEC is pretty efficient, I would say, more efficient than ultracentrifugation, for example. Mm-hmm. So it's, for me, it's a bit strange that then, you see such a low efficiency while, normally you get a really high efficiency with SEC. from different types of plasma?
I don't know.
Or could it be, for example, that in vivo maybe EVs are coated with other proteins and these would interrupt the interaction with
exactly
LDLs?
where the phenomenon of the protein corona or any corona on the top of EV structure is out there, but it's very difficult to determine because I mean, different isolation methods and so on may compromise the natural coating that happens on top of EV's membrane. So
yeah, it's difficult to prove this in vivo. That's true. No, but again, nice work. Thank you.
Thank you.
Okay, next question from Leon. Would you like to unmute yourself?
Sure, great talk. I was just wondering if you've looked at if the EV is associated with apolipoprotein E as well. This is abundant in brain and microglia. Can pretty much directly take that out.
No, we didn't look at ApoE, just ApoA1 and ApoB.
The next question, Anirban.
Great talk. The tumor microenvironment does play a role. Like if the EVs are coming out, like is there absorbing it first and then it's letting it into the bloodstream? So is there any role of it? What do you think?
Which microenvironment are you talking about? The blood?
The tumor microenvironment.
I think that the tumor microenvironment and the tumor cells may play a role, of course, in the EV structure and composition. Not sure if it may play a role in their interaction with circulating.
And the immune. Right.
The next question is like follow-up about previous discussion. Jack, would you like to clear up a little bit?
Yeah, I just wondering whether this is the illusion with LDL will be such as clogged or make it the illusion much slower and so on. And so your EV, the less efficient, maybe it's not because they're interacting with LDL, maybe just LDL simply making the whole, anything that's smaller vesicle will be decreased. So let's say you choose other non-EV particle, do they also decrease? I mean, if that means it's a non-specific effect, no interaction.
I think that this may be answered just looking at the AFM morphology of the samples, and that the fact that the AFM shows two different types of interaction based on the type of metastatic EVs that we choose. And also the different behavior of the two samples if incubated with LDL or HDL and equally run by through SEC column. So I don't think that this is that unspecific that it may happen with all the nanoparticle out there. We could test with synthetic liposome to see if this happens or not, but since we got the AFM results and the HDL results, we didn't think about it.
The next couple of questions from Pramesh.
Yeah, hi, great presentation, Sarah. This is Ramesh from Oklahoma. This is the first time I'm participating in this. It's great work, and thanks to Ken. My question was two ways. One was if LDL is really playing a role in metastatic nation, maybe in establishing metastasis in the brain or any other organ. So have you thought about it potentially serving as a marker? If you take the exosomes from my patient, say stage 1 patient, and look at the content, would you be able to tell it could be a predictive marker for potential for metastasis in that particular individual? That's one. And the two was, have you thought about looking at different stages of, say, it doesn't matter which disease, breast cancer, melanoma, or lung cancer. If you take exosomes from different stage of the disease, do you expect there's a different level of LDL bound to the?
Well, these are both interesting questions. So about the first one, whether they can be used and validated as biomarker, I think that once this hypothesis will be further proved with in vivo studies, so this relation will be confirmed, any biological particle and every component of the biological particles involved in a pathological process may work and may be validated as a biomarker. So I think that is a good hypothesis. About the variation based on the stage of the pathology, I think that they may absolutely vary. Also because with the progression of the brain metastasis, well, at the earliest stage where there is not a brain metastasis, but the metabolism and the primary tumors is working in order to create a favorable microenvironment, the BBB is still intact, right? And the permeability is still low, so they have to hijack its transport in order to get into the brain parenchyma. If we take into consideration advanced and formed brain metastasis, the situation changed completely because we have leaky BBB, and also the transport are completely different, and the brain tumors may recruit more lipoproteins and particles of that kind in order to fuel itself. So there may be absolutely a difference between earlier and late stages.
We have next question from Estefania. Very long
Hi,
question.
hello.
Hi.
Thank you for the nice presentation. I think it's a very interesting work. And I was wondering about the ratio of EVs and LDLs that you use for the co-incubation of these particles, because we know that in blood plasma, for example, LDLs are far more abundant than the EVs. So if you try to make a physiological ratio for your experiments, that's one question.
Yeah. So I agree that lipoproteins are far more abundant than EVs in the blood. So unfortunately, the LDL are too small to be reliably, to make a reliable quantification by NTA. So I went by protein quantification, although I don't like very much the approach, but was the only one that allows me to work in order of magnitude in order to reproduce something similar that we may find in a biological sample. So let me see, because I took notes of it, because I thought that it could be a potential question. So I used 500 micrograms of lipoproteins and 10 to the 10 EVs, which are 10-fold more or less less micrograms in protein.
Okay. Great. So thank you very much for the precise number for the ratio. And then I was also wondering if you try to see this, because I think it's very interesting that you found different interactions between LDL and HDL. And if you try to also look into the other low density lipoproteins, like the VLDLs or the chylomicrons.
No, I didn't, but mainly because it's the VLDL and chylomicrons is more a transition state that you can find postprandial or while they are transforming to become then LDL during the lipid transport. It's not a, let's say it's not a stable state, so they are difficult to find. And we, as this is a proof of concept study, we decided to go for commercially available sample. I think that if you want to find VLDL, to study VLDL or chylomicrons, you have to talk with clinicians or clinical lab and or make it a synthetic version yourself.
Okay, thank you. And one final question that I'm just curious if you try to do the cryo-EM after the incubation of these particles, and if you were able to see these interactions by cryo-EM.
Actually, that was one of our points, but COVID happened, so we could do just the AFM that was in a lab very close to us. The cryo-EM collaborator, he's absolutely amazing, but they work in Israel, so it was a little bit difficult.
Okay, let's try another one. We have like a method question from Marka. How did you analyze CD63 expression after the LDL EV mixing experiment? So which samples prep has been performed and how did you detect CD63?
We detected CD63 using an ELISA assay.
And another...
Yeah, but maybe, so the question was really that, like, what did you do afterwards? So you have your LDL and your EVs and then you incubate them together. So what was your preparation afterwards before you really conclude that you could not detect any more CD63 because of masking?
It was the sample mixed, incubated at 36 degrees for a couple of hours, and then we tested the different dilution of it, and in all of them, we couldn't detect CD63. And we decided to then analyze the dilution that, the max dilution, as even though increasing the dilution, we couldn't detect it, and to hypothesize the masking.
Okay, thanks. Very nice talk. Thank you.
Thank you.
We have a couple minutes. So have you analyzed or plan to analyze glycan-glycan or glycan-protein interactions in EV's LDL association?
No, we are working on the Dr. Wolfram lab, when I was there as a postdoc, and also now I know that they are working on EV glycans and how they play a role in the interaction with targets that may be circulating particles and cells. So I suggest to email the other co-corresponding author of the paper, and I'm sure that she will update you about this topic.
Okay, and going back to Alexander's question, since he cannot ask, how is the interaction mediated between EVs and LDLs? Is the LDL receptor present on EVs or is it an ionic interaction?
So this is a very interesting question. We had the same doubt whether EVs express LDL receptor. So we tried to, but of course we wanted also a sensitive method that allowed the comparison of the LDL receptor quantity between regular EVs and brain-seeking EVs. We tried a few ELISA assays while I were in the lab without having very reliable results, meaning that we could detect some LDL receptor in the samples, but the difference and the pattern between the samples were not consistent. So we decided to maybe opt for other techniques. I don't know whether the lab is currently working on mass spec or any other type of techniques in order to determine whether and in which quantity the two EV samples are expressing the LDL receptor.
Okay. And have you titrated the EVs with LDL to determine concentration dependent on the interaction?
No, we didn't do titration.
Okay, I think that's all question. I think, Ken, you also had like question?
Well, I did have a question, but it's already been addressed. So
okay.
So thanks everybody for all of the good questions and comments.
Yeah. Thank you, Sara. Great presentation.
Oh, thank you, and thank you for the chance of discussing this with all of you. I had very insightful question and inputs and feedback, and I appreciate that.
All right. Well, thanks everybody for joining today, and we look forward to seeing you again very soon.
Thank you. Bye.